time, all of you have gone through the new reduced syllabus of biology. So mainly in the syllabus, so let's discuss a few things. We used to have 6 units. In last class, we are dealing with the last unit, right? We are dealing with diseases, position control, health, the key to life, and we are talking about hygiene. Clear? So it was under the unit 6. But now, when we look into the new reduced syllabus, the unit 6 has been removed. Yeah? So, when we look into the syllabus, you find that we have got the first unit, basic biology, where we have got the cell and the tissues. We have got second unit, you can see over here, the flowering plants, where we have learned about the structure of flower, we have learned about pollination, self-pollination, cross-pollination, and we have also learned about fertilization. In unit 3 is plant physiology, where we have learned about the seed structure, right? Where we have learned about the monocot seed, we have learned about the dicot seed. So today we will mainly learn about the structure of a seed and its germination under the unit 3, plant physiology. Then we have got unit 4, diversity in living organisms, where we have to only go with the economic importance of bacteria and the fungi. Yeah, so that wanted to go with the 5 inner classifications. We have to go through only the economic importance of bacteria and the fungi. Then we have got unit number 5, human anatomy and physiology, where we have to learn about nutrition, right? We have to learn about the structure of a tooth, then digestive system, skeleton, and structure and function of skin. So overall, the unit 6 has been removed from here, new reduced syllabus. Clear? So there is no need to go to the last unit. So now today we will begin with a new chapter, with a new unit under the plant physiology, and the name of the chapter is seeds, structure and germination. So before going into the structure of the seed and the germination process of the seeds, we will first learn about the actual meaning of seed, fruit and the grain. So what do you mean by fruit, what do you mean by seeds, and what do you mean by grains? Okay, so they are the main things which we will learn today. So what is the actual meaning of fruit, seeds and grains? So what do you mean by fruit? So fruits are actually the ripened ovary. Now over here, try to recall the process of fertilization which we have done earlier in our classes. Fertilization is the process of fusion of haploid male gamete and the female gamete. So we have learned this thing here. Now in case of plants, very carefully, in case of flowering plants, we have learned the process of double fertilization, where there will be formation of a zygote and the formation of an endosperm. We have learned that thing. Clear? And what are the actual changes that occurs after fertilization? We have also learned that thing. Post-fertilization changes in a flower. So under the topic post-fertilization changes in a flower, we have learned that after fertilization, the ovary will be seeds, ovary into fruits. Keep in your mind. So what is a fruit? So fruit is actually a ripened ovary. So after fertilization, ovary will change into a fruit. So fruit is the enlarged ripened ovary. Clear? And the ovarian wall forming the fruit wall encloses the seed. So inside the fruit wall we will find the seeds. And what is the function of fruit? The fruit protects the seeds and help in seed dispersion. Now, what are the different parts of a fruit wall? So when we look into the structure of the fruit, we will find 3 layers: epicarp, mesocarp and the endocarp. These are the fruit wall, and this particular fruit wall will enclose the seed. So what is a fruit? It's actually a ripened ovary, and the ovarian wall which forms the fruit wall encloses the seeds, and the function of the fruit is to protect the seeds and help in their dispersion so that it can germinate, it can multiply. Clear? So you can see over here the different types of fruits and the structure of typical fruit with its fruit wall. The next one, what do you mean by grain? A grain is actually a fruit in which the fruit wall and the seed coat are fused together to form a protective layer. One more time, grain is actually a fruit, but in this case, the fruit wall and the seed coat are fused to form a protective layer. Now, just now we have seen the structure of a fruit wall. What does the structure of a seed coat looks like? Look over here. So you will find the outermost layer, and this layer is called the seed coat. So when we go in detail, we will find the structure like a star segment. Clear? So grains are actually the fruit in which the fruit wall and the seed coat are fused to form a protective layer. Example you can see over here, image grain and the grain. Now, coming to the most important point, what we mean by seed. So, after fertilization, we have learned that the ovary will change into fruit, whereas the ovule, will change into a seed. So, seed is the ripening ovule. So, after fertilization, the ovule will develop into a seed. Now, seed, it contains an embryo which develops into a new plant. So, when we look into the seed structure, we will find that the seed contains an embryo, and that embryo will germinate into a new plant. Now, seed coat protects the embryo from the mechanical damage. The outermost coat, we call it seed coat, it will protect the embryo from mechanical damage. Example are bean seeds and the pea seeds. So if you look over here, the structure of a typical seed, you will find the micropyline over here. So the seed will germinate, okay? And after germination, you can see over the structure, you will find the cotyledons. There is a diploid, there are two cotyledons, you will see it afterwards. Then you will find these structures coming out, radicle and the plumule, P-L-U-M-U-L-E. Plumule means the future shoot, S-H-O-O-T, shoot of a plant. And radicle, R-A-D-I-C-L-E, radicle means the future root, R-O-O-T, root of a plant. Clear? So seed is actually a ripened ovule which contains an embryo, and that embryo will germinate into a new plant. Clear? And the seed coat will protect the embryo from mechanical damage. Now, going in detail about the seeds. Seed, it's a mature ovule after fertilization. So you have learned that after fertilization is over, the ovule will develop into seeds. Right, number 2 point, it also contains a tiny living plant called the embryo. Clear? Now, embryo is mainly formed by the fusion of sperm nucleus and the egg nucleus. So now, recall the process of double fertilization. Clear? Within the egg apparatus or in the embryo cell, you have observed that there will be 8 cells. 3 cells at the genital end or the anteporal sides, 3 cells at the metaboral end, and 2 at the center. You remember, right? We read that. Clear? So whenever the pollen grain reaches the stigma of a flower, it will germinate. It will germinate and it forms a pollen tube. The pollen tube develops through its stance and enters into the embryo cell from the micropylar region. And within the pollen tube, 2 sperm nucleus or 2 male gametes will be released. All the things we have discussed already. So try to recall it back. So one of these sperm nucleus will go and fuse with the egg, which forms the zygote. The zygote will develop into an embryo, and this embryo will germinate into a new plant. The second male gamete will go and fuse with the central polar nuclei cell and will form a endosperm. Clear? So all these things we have discussed already. So try to recall it back. Clear? So seeds contains a tiny living plant, the embryo, which is formed by the fusion of the sperm nucleus and the egg nucleus. So this is the male gamete and this is the female gamete. You will form a zygote, and zygote will develop into embryo, and whenever the embryo will germinate, it will develop into a new plant. The embryo consists of radicle, which is the future root, and the plumule, which is the future shoot, and the cotyledons or the seed leaves, which are the future leaves. That means if you look over here, so this is the structure of a particular seed. It is mainly composed of cotyledon, which is composed of plumule, and which is composed of radicle. So this is the particular structure of a seed. It is composed of plumule, the future seed, shoot, radicle, the future root, and the cotyledon. Clear? Next one. The embryo remains in an inactive state, but upon exposure to favorable conditions, it germinates. That means a particular seed will only germinate if we get favorable conditions for germination. If the conditions for germination are not favorable, then it will not germinate and will enter into an inactive stage, which is called a dormant stage. Yeah, so dormant stage means the inactive stage of a seed where it will not germinate. But those inactive seeds or the dormant seeds will germinate if the conditions for germination will be favorable. Next one. The seed may also contain the endosperm, which is the food material for the nourishment of the embryo during germination. So hope you have got a thing that the second sperm nucleus or the male gamete will go and fuse with the central polar nuclear cell and forms the endosperm, which is the rich of food material for the developing embryo. Clear? So over here you have to try to recall the process of double fertilization in flowering plants, which we have discussed under the topic or the chapter pollination and fertilization. So go through it and it will be clear for you. Now, next one. We learn that the seeds can withstand unfavorable conditions of temperature. Some seeds are known to remain dormant even up to 100 years or more. Meaning is that for the germination of a seed, the conditions must be favorable. The temperature, the moisture content in the soil, alright? The salinity of the soil, all of these factors like temperatures, weather, or the climatic conditions, all these factors will determine or will affect the process of seed germination. Alright, so if these particular conditions will not be favorable for seed germination, then those seeds will undergo a stage of inactive, which is called dormant stage or dormancy, where they will not germinate. So that's why most of the seeds, they should have the dormancy period of up to 100 years or more. But if the conditions become favorable, if the temperatures become favorable for germination, clear, then the seeds will overcome the steps of dormancy and they will germinate. Clear? Now next one. Classification of seeds. Classification and structure of seeds. So we have got different shape and the size of the seeds. So that means seeds is to vary in size. So some of them are small, some of them are quite large, and some of them are large. So we'll see the smallest seeds, alright, we'll see the quite large seeds and the largest seeds. So small seeds are barely visible to our eyes. Example, you can see the poppy seeds, strawberry seeds, which are quite very small. There are some quite large seeds like the mango seeds, plum seeds, and some of the largest seeds like the coconut and the double coconut. So the size and the structure of the seeds of different plants vary considerably, but the basic structure of the seeds is the same. So saying that the size of the seeds can be different. It may be small, quite large, or the largest one, which you have seen. But the general structure of most of the seeds used to be same. So keep in your mind, size may vary, size may be different, but the general structure of most of the seeds used to be same, used to be similar. So over here you can see the smallest seeds, the poppy seeds, you can see the quite large seeds, the seeds of the mango, and you can see the coconut, which is the largest one. So we have got different sizes of seeds But keep in your mind, though these sizes are different, but the general structure of the seeds used to be same. Next one, the type of seeds depending on the basis of endosperm, or we can say depending on the presence or absence of endosperm. Remember, endosperm contains the richer food material for the germinating or developing embryo. So during the germination or development, embryo will get its nourishment from the endosperm. Clear? So that means the endosperm will be utilized completely during germination, or it will not be utilized during germination. Clear? So on that basis, we have got two types of seeds: albuminous seeds and the ex-albuminous seeds. Keep in mind. Clear? Albuminous seeds is also called endospermic seed, and ex-albuminous seed is also called non-endospermic seed. So over here you have to keep in mind that this particular differentiation has been done on the basis of presence or absence of endosperm during the seed germination. So what do you mean by albuminous seeds or the endospermic seeds? So in such seeds, cotyledons are thin, they are membranous, and the endosperm persists. That means endosperms are not completely consumed during the seed germination, or a little bit of remaining parts of the endosperm will be there. Okay, so over here, what are albuminous seeds or endospermic seeds? So endosperm will persist over there even after the germination is over. Cotyledons are thin and membranous. So example, monocot albuminous seeds are cereals, okay, millets, dicot seeds you will find coffee, custard, apple. Now number two, ex-albuminous or non-endospermic, where the endosperm does not persist. They are completely consumed. So in such cases, the cotyledon stores the food. So over here, cotyledons will store the food material. They become thin and fleshy. In case of albuminous seeds, cotyledons were thin, but in case of ex-albuminous, cotyledons are thick. Clear? Now, next thing over here, endosperm persists. That means endosperm will provide the nourishment of the food material, but over here, the cotyledons becomes thick and fleshy, and it will store the food. So example over here, in case of ex-albuminous, monocot seeds you will find orchid, okay, amaryllis. Dicot seeds you will find the gram, pea, mango, and stuff. So you can see over here, the two particular diagrams will be over here. The structure of a maize grain, you can see. The remaining part of the endosperm. Okay, so it is the endospermic seeds where we will find remnants of endosperm. Whereas over here we will find the cotyledon part which will store the food material. So this is the non-endospermic seeds where the cotyledons store the food material and becomes thick and fleshy. So over here the structure of endospermic seeds and the example of non-endospermic seeds. So kindly go through it. The next one, broadly, the seeds are two types depending on the presence of number of cotyledons, monocotyledonous and dicotyledonous. So we have got two types of seeds depending on the number of cotyledons. Clear? So they are monocotyledonous and dicotyledonous. Monocotyledonous means it contains only one cotyledon, because the term mono means one. So in case of monocotyledonous, there is presence of a single cotyledon. So cotyledonous means cotyledon, the future leaf of a seed or a plant. Dicotyledonous, the word di means two, and cotyledonous means cotyledon. So in case of dicotyledonous, there will be two cotyledons. So on the basis of presence of number of cotyledons, we have got two types of seeds: monocotyledonous seeds and dicotyledonous seeds. Monocotyledon sheets contain single cotyledon, example maize, grasses, and dicotyledonous contain two cotyledons, example pea, and bean. So now in the next class, we will look into the structure of a monocot sheet and the dicot sheet. So this is much for today. Continue in the next class and kindly go through the new reduced syllabus of biology. Thank you and see you in the next class.